we have carbohydrates, we have proteins, and we have nucleic acids as DNA or RNA. Okay. So today we will discuss one of the component we have is protein, and the basic unit of those protein is amino acids. Okay. And there are many number of main amino acids we have in our body, and those all form a polymer and fold like primary, secondary, tertiary structure to give you a functional unit which is called a protein. Okay. So you might have heard of some of the protein you have in your body. An example of protein you can give? Yeah. Your hair is also made up of protein. I think you might have seen those on advertisements, keratin protein, keratin rich, conditioner, shampoo, or anything else. So mostly your component or mostly structural framework of your body is made up of protein. Okay. And the component like units of those protein is amino acids. So we'll discuss what are proteins made up of, structure of amino acids, which is very hard to remember. I struggle a lot in my undergrad days just to remember the structures of those protein. Okay, but I can promise you at the end of this class you will be able to remember all 21 amino acids with structure and name. Okay, I think I hold my promise, and thank you for giving me the opportunity. And we'll discuss about peptides, primary structure, secondary structure, and tertiary structure, and then about the proteins, how proteins are folded in different forms, and what are the different form of protein. Okay, so starting with the very first slide, introduction to amino acid. So this chapter includes, we will discuss about the structure and naming of amino acids. We'll discuss how different amino acids are structurally different in terms of their R group. The second we will discuss is about structure and properties of the peptides. As you know, amino acids polymerize to form peptides, right? And third is ionization behavior of amino acids. You might have seen amino acid reacts differently when they are present in different pH. Okay, and we will learn about isoelectric point and deuterium. Okay. After that, we will discuss mostly about method of characterization of peptides and protein. I will cover only 3 parts, and 4th part will be covered by Dr. Narayan. Okay, so we'll focus mostly on structure and naming of amino acids, properties of amino acids, and how to calculate pi value, which is isoelectric point. Okay, starting with the very first introduction, how amino acid perform different tasks in our body. First is catalysis. Okay, catalysis word is derived from, mostly chemistry people use catalysis, right? Catalyst. Okay, so catalysis means you need a catalyst if you are talking in terms of chemistry, right? We need catalyst for reaction to go faster. So your body also needs catalyst in order to fast the reaction that is happening in your body. So imagine you are breathing oxygen, it goes into your lungs and then goes into tissues, right? This could be a very slow process if there is no biological catalyst in your body. Okay, and the biological catalysts in your body are called anyone? Biological catalyst of your body? Enzymes. Okay, enzymes helps in catalysis in your body, and different types of enzymes are called they are all part of protein. Most of the enzymes are proteinaceous in nature. Okay, except a few, which is ribozyme or some other enzymes. Okay, so we have enolase, which is present in glycolytic pathway, and you might have heard about glycolysis when we break down carbohydrates, complex carbohydrate to simpler ones and derive ATP, which is energy currency of your cell, right? So these all enzymes fast those reactions, which helps to generate more and more ATP, hence energy in your body. Okay, so these biological enzymes are important, and they are in category of proteins. They are also responsible for transport of molecules. If you talk about a very good example, which is hemoglobin, they are excellent transporter of gas. Okay, mostly CO2 and oxygen, right? So you might have understood the importance of protein in our body, since almost every process is having the involvement of those proteins. Third is structure. Most of your membranous structures are protein, and they help in transport of molecules across the membrane. Okay, so if you have eaten something or a drug that needs to go inside the cell in order to do its function, right? And transport of those drugs or any molecule outside the cell inside towards the cell could be done by any membrane protein you have. Okay. Also motion. Motion means muscle movement, right? You eat a lot of protein if you go to a gym or want to build muscles, right? So why you eat protein? Because those proteins are gonna made up with your muscles, right? This comes under actin and myosin, which is muscle tissue. Okay, that comes the whole story if you want to go ahead with that. So let's talk about amino acid, which are building blocks of protein, and they are heteropolymers of amino acids. There could be two different terms, homopolymer or heteropolymer. Homo means same, right? And hetero means different. So if we talk about homopolymers, all the units will be same, and the other one is heteropolymer. So homopolymers and heteropolymers. Okay, homo word means same, and polymer means polymers of units. If there is a heteropolymer, it means we have different units in your protein. Okay, and by different we call protein as heteropolymers because they have different units in them, and differences is just because of their R group, and we'll see how these R group are different, and we'll try to draw the structures of those R groups. Okay, they are very independent molecule, and they have biological functions like they have capacity to polymerase, they have specific acid-base properties due to their R chain, they also have physical properties like melting point, and they also have chemical functionality. Chemical functionality means they are capable of doing a function in your body, right? Most of the proteinaceae they have some function in your body, that's why they are there, right? So let's start about amino acids. Okay, amino acids could be alpha amino acid or beta amino acid based on how they are attached to your functional group, which is carboxylic group. Okay, so let's discuss about that. I'll be more comfortable with whiteboard and marker. Okay, so maybe I'll switch a few times, and you have to follow where I'm switching. So you see, this carbon can be called as a chiral carbon. I think you might have learned about a chiral carbon, which means basically if you have a carbon, and this carbon is surrounded by 4 different functional groups. Okay, A, B, C, and D. By A, B, C, D meaning different, I mean they have different functional groups. Okay, and if they are different, which is A equal not B is not equal to C is not equal to D, we can call this carbon as chiral carbon. Okay, this carbon is chiral carbon. Okay, so chirality means they have non-superimposable mirror images. Okay, that calls enantiomer and so other things later on. So this carbon is called chiral carbon. In here we call this carbon as chiral carbon, and they have optical properties differently. So it is attached by hydrogen, carboxyl group, amine group, and R group. This R group varies differently. Okay, so this R group could be anything. Okay, it could be simplest from H to complex like tyrosine or phenylalanine. Okay, so this R group you have to remember for each one. Okay, and these 3 groups will be same for all the amino acids. So the only thing you have to remember is what is in position of R, which is alkyl group. Okay, so this carbon is designated as alpha carbon. Okay, why? Because this is directly attached with carboxyl group. Okay, directly attached with carboxyl group, and that's why they are called as alpha carbon. The other reason why these all amino acids are called alpha amino acids, there could be beta or gamma amino acids, and I can introduce you to later on. Okay, so I think up till now we are familiar with alpha carbon. There could be another good question here. What is alpha hydrogen? Okay, anything connected to any hydrogen connected directly to alpha carbon is called alpha hydrogen. Can we call this hydrogen as alpha hydrogen? Yes, no. No, because they are not directly connected to alpha carbon. Okay? I can give you a few examples to understand what does alpha carbon means. Can we call this as alpha hydrogen? Suppose this is alpha carbon. Okay? How many alpha hydrogens are there? Anyone? How many alpha hydrogens are there? No one. Zero, right? How many alpha hydrogen are there? One. How many alpha hydrogens are there? Three, right? Okay, so apart from that, let's see, this is attached with an H2, right? This is why we got this alpha amino acid. This is R group, okay? This is alpha carbon, and amine group is attached with alpha carbon, and that's why we call this as alpha amino acids, okay? Let's draw another structure to understand. If I draw carbon, COOH, hydrogen, carbon, can we call this alpha amino acid? This is functional group FN, okay? This is carbon alpha, okay? And this is carbon beta. And amine group is attached with beta carbon, okay? That's why we call this as beta amino acids, okay? Can I introduce a new carbon here and call this as gamma amino acid? Yes or no? So depending upon number of carbons you have in between, you can call this alpha carbon, beta carbon, or gamma carbon, and depending upon that you can call this as alpha amino acid, beta amino acid, or gamma amino acid, okay? Let's move on forward and understand how these amino acids have a chirality, okay? By chirality, I mean they have a chiral carbon, and I already discussed about chiral carbon. A carbon is chiral when they have 4 different functional groups attached to them, okay? If we talk about the simplest amino acid we have, which is glycine, it is achiral. By achiral means you can replace this molecule, which is CH3, to H, okay? Now all 4 different groups are not different anymore, okay? If you replace this by H, there is another H, which is hydrogen, right? And there are 2 hydrogen. So this is not fulfilled in that case. A equals B equals C. A not equals to B, not equals to C, but equals to D, or anything. I think you have, if you have 2 similar groups, you cannot call that chiral as chiral carbon, okay? So the example, all amino acids are chiral except glycine. Reason why? Because glycine has a structure like this, okay? This is structure of glycine. COOH, this is hydrogen, this is NH3, and this is hydrogen, okay? Can you see this? Hydrogen equals hydrogen, so this is not chiral, okay? This is only amino acid which is not chiral. It could be a good question in your quiz, right? So amino acid which is not chiral is glycine. Why? Because they have 2 similar groups, which is hydrogen, right? Let's move to see how amino acids are characterized into D amino acids and L amino acids, okay? So D and L are 2 different form of enantiomers, okay? If you have idea, enantiomers are isomers, okay? Which non-superimposable mirror images, right? Like my hand, okay? My hand in the same side cannot superimpose. Can you see that? Right? It's not superimposable. This way it's superimposable, right? So if you compare these two structures, is it superimposable? No. But they are mirror images. Is it also? They are mirror images of each other, but they are not superimposable. This is a mirror image. Can you see that? If you place a mirror in between, these two structures will be same, right? But they are not superimposable. Superimposable means putting one after the other, right? So enantiomers are isomers with mirror images, but not superimposable with each other, right? And these are example of T and alanine. Okay, we'll discuss about the structures of amino acid later on. I think you might already know about the meaning of these dash and wedge structures. Okay, this is above the plane and this is below the plane, right? So this is a 3D molecule. Okay, if you imagine these hydrogen and ammonium NH3 group, they are above the plane, okay, and these two are below the plane, and this is a plane of paper where carbon is there, right? So this is how we visualize 3D structures. Okay, this is more chemistry than biology. Okay, this is the most difficult part, and this is a very important thing to learn about structures of the amino acids. Okay, it is categorized into 5 groups. First is aliphatic non-polar, aromatic, polar, positively charged or negatively charged. Okay, these two also are called basic amino acids and acidic amino acids. Okay, so let's understand everything and then we'll categorize one by one. Okay, you will be able to learn the categories, how they are categorized once you learn how they are having the different R groups. Okay, the first category we'll discuss is non-polar aliphatic groups. Okay, there is a very common term in chemistry or biology we call polar or polarity, right? Polar means they have two poles, like magnets has two poles, north pole and south pole, right? And molecule also has two polarity, having a positive charge or negative charge, maybe a partial positive charge or partial negative charge depending upon the molecule. Okay, so none of them are polar. Bipolar means, a very good example of polar molecule is, anyone who can provide me a good example of polar molecule?
Water.
Very good example. Excellent example is water, which is H2O. Okay, if you write the structure of H2O, it looks like this with a bent shape. Okay, water molecule has a bent shape, and oxygen and hydrogen having different electronegativity, because it means they have different capacity to pull electrons towards themselves, right? Oxygen has higher capacity to pull electrons towards them, and that's why they are partial negative charge. Okay, and hydrogen has partial positive charge. This positive and negative charge in the same molecule make this molecule as polar molecule. Okay, so water is a polar solvent. It's an excellent solvent, right? Universal solvent. So this polar will indicate here, because there is no polarity, though there is no much electronegativity difference between two atoms in R groups. That's why they are categorized as non-polar groups. Aliphatic means they have simple chain of carbon. Okay, by simple chain means I mean carbon carbon, or maybe carbon carbon carbon, but they don't have any ring or any aromaticity. Okay, that's why they are categorized as non-polar aromatic aliphatic R groups. Okay, aliphatic and aromatic are two different categories of carbon chain. Okay, could be a cycle, but it could never be an aromatic ring. Okay, so let's remember this as a whole. Okay, so we have glycine, alanine, proline, valine, leucine, isoleucine, and methionine. Okay, from now on you have to focus more on class, since I will try to imagine structures in the air. Okay, so the first thing you have to remember, once comes non-polar aliphatic group, you have to remember how many amino acids are there. Okay, and you have to immediately think of how many structures do you have to remember. Right, I have a simple mnemonics to understand or to learn. Okay, you might remember this structure at the end of the class, but when you go to like write paper or write exam, might not be able to recognize those structures and write it down. Okay, so I tried to make a mnemonics so that you can remember all the structures. Okay, and it is like every time Apple launches a new iPhone, right? You said I need an iPhone, right? You need iPhone 16 Pro Max, that's certain. So it says, give me Apple. Okay, give me Apple. Okay, let's remember the name of the amino acid they constitute, right? The first one is G for glycine, I for isoleucine, V for valine, M for methionine, A for alanine, P for proline, and L for leucine. Can you remember the name of, at least name? Okay, now we are moving step by step. Okay, first we will remember the names and then structures. Okay, so the first group will be remembered as give me Apple. Okay, G for glycine, I for V for, you have to speak up. That's a way how you create your memory. Okay, if you don't speak up, you will not remember those names. So please speak up with me. Okay, G for, I for, V for, M for, A for, P for, L for, one more time, G for, I for, V for, M for, A for, P for, and L for. Okay, that's it. So I think we are at least remembering the name. Okay, I will repeat one more time. Okay, G for, I for, V for, M for, A for, P for, L for. Okay, that's clear. Now we have to remember the structure. Okay, I'll move forward one by one. The simplest amino acid we have is glycine, right? Glycine has simplest could be hydrogen, right? And then we can move forward with alkyl groups, which is methane, ethane, propane, butane, and so on. Okay, so the simplest we have is glycine. Okay, so I don't know how to move ahead with this whole thing. Okay, I like to teach more on whiteboard. So I will skip that for one time so that you can remember the names. Okay, so yeah, let's see how we remember the names of the amino acids. Okay. No worries. Do we have any spare marker? Anyone? Great, thank you so much. So let's understand how we remember the name of the structures we have. Okay. So as we said, Z for glycine. One more time. I for isoleucine. V for valine. M for methionine. Okay. A for alanine. P for proline. And L for leucine. Okay. There is no PPT, so you have to remember those structures now, right? So glycine is simplest amino acid. Okay. The first thing we'll do is I'll draw the backbone of the amino acid you have to remember. Okay. And now I'll put different R groups just to remember the names. Okay. Clear? So let's go one by one. So I think other groups are very common, right? Other groups are common. The common you have is a carboxyl group. You have a hydrogen. You have a NH3, okay, or NH2. Repeat NH2 if they are not ionized, right? So we have to remember everything is common except the R group, okay? And let's draw everything. You have to draw with me, okay? That's how you remember things. The first R group is glycine, and which is simplest. And glycine has a hydrogen, which is H, right? So we'll remember glycine first, and there's an order how we remember, okay? And second is alanine. So first is glycine, second is alanine. So this is glycine. Alanine is second simplest amino acid. First simplest is glycine, second simplest is alanine, and alanine has a CH3. That's it. Glycine has hydrogen and alanine has CH3 group. Okay, so we'll write down CH3, and this is glycine and this is alanine. Let's remember the third one. Okay, third one is let's start with leucine. Okay, and then we'll discuss about isoleucine and then valine. Okay, is this leucine? Yes or no? It has 4 carbons? No. No? Leucine has something different. The last one, okay? Last one, CH3 and CH3, okay? And this is isoleucine. Okay, the way how you remember it is both of them has 4 carbon. Okay? This is 4 carbon and this is also 4 carbon. Can you see a pattern in their name? Is it correct? It's otherwise? Okay. So this one is leucine, right? And this one is isoleucine. Okay. Thank you. So there's a pattern, no? What's wrong? Isoleucine is isomer of leucine. Leucine is CH2. Come on, man, he was right. So this is leucine, right? Okay. And this one is, I was correct in the beginning, isoleucine. Okay. So leucine. What is the other thing we have is iso. Iso means isomer. Okay. So leucine is isoleucine is isomer of leucine. Okay. And leucine has 4 carbon. So the way you have to remember is 1, 2, 3, 4. Okay. And isoleucine is isomer of leucine. So you have to put this thing over here. Okay. And this is isoleucine, right? I think I'm correct. Don't get confused. This is isoleucine and this is leucine. Okay. So up till now we remember glycine, alanine, leucine, and isoleucine. Now we have to remember valine and proline. Okay. So let's discuss valine. Can you see V? V for valine. Okay. Let's invert the structure. Okay. By inverting, I mean V could be this V. Okay. And this V could be CH2. Write it CH, CH3, and CH3. Can you see this is V? Right? This is inverted V. Inverted V is valine, right? So to remember, you have to invert the structure, and this is how you write in their bond form, right? Valine, okay? This is valine, and this is leucine, okay? This is isoleucine, okay? So you have to remember those structures in your mind, okay? So you don't have to go through all those things. So let's start with the beginning, okay? Glycine is simplest is H, okay? And then alanine is CH3. Then leucine 4 carbon, okay? 4 carbon is 1, 2, 3 and 4. Isoleucine is isomer of leucine. So you will shift this over here, okay? And this is isoleucine, okay? Glycine H, alanine, come on, CH3, leucine 4 carbon, 1, 2, 3, 4, right? Isoleucine is isomer of leucine, okay? Valine is opposite V, inverted V, okay? CH, CH3, CH3, okay? And then last one is proline. Proline is slightly different from other amino acids because it is cyclic in structure, okay? So this is how we represent proline, okay? CH2, 1, 2, 3, 4, 5. It's a 4 ring or 5 ring? 5 ring? 1, 2, 3, 4. It's 4 ring, okay? So you have to remember number 4 is important. You see that number 4 is important. Number 4 is important in remembering the names of aliphatic non-polar amino acids, okay? Okay, I'll revise one more time to remember all the names. Let's start with glycine. Glycine has simplest H. Alanine, CH3. Then leucine, 4 carbon, 1, 2, 3, 4, right? 1, 2, 3, 4, right? 1, 2, 3, 4. And isoleucine is CH, 1, 2, 3, 4, right? 1, 2, 3 or 4, right? And then we have glycine, alanine, leucine, isoleucine, valine. Valine is opposite V, right? CH, CH3, CH3, right? And then lastly we have proline, which is again 4. 1, 2, 3, 4. 4 ring, okay? So there is a similar structure we learned tryptophan, which has 5 ring, okay? And it's confusing from this, okay? So you have to remember how they are made, right? So I think we are familiar now with the first common you have, which is, let's understand the structure. Okay, you remember this? First is glycine, hydrogen, second is alanine, CH3. Then we'll learn leucine first. Leucine is 1, 2, 3 and 4. And then isoleucine is isomer of leucine. Write this over here. This is 1, 2, 3, 4, right? This is isoleucine. Then we'll learn about valine. Valine is opposite V, okay? And then proline. Proline is 1, 2, 3, 4. 4 ring, okay? The one we missed is methionine, okay? Can you see this structure? Methionine is not common, but it has also 4 numbers. 1, 2, 3 and 4. But it contains S. S means sulfur. So this is one of the sulfur containing amino acid. Write on your notebook. Sulfur containing amino acid is methionine, okay? And there is another one, cysteine. We'll learn after maybe a few slides, okay? So 1, 2, 3, 4, and in third position we have sulfur, okay? So this is how you remember. I think you remember glycine, which is H, alanine, CH3, valine, 1, 2, 3, opposite B, and then proline, cyclic, and then leucine is 1, 2, 3, 4, isoleucine is 1, 2, 3, 4, right? Isomer of leucine. And then methionine is 1, 2, 3, 4, but in third position, in place of carbon, it's sulfur, okay? And this is how evolution of amino acid happens, right? We'll see how this amino acid is very common to one of the amino acids we have in next slide, okay? Then after, let's move to aromatic amino acids, okay? It's a very simple form. The hardest was that to remember, okay? Because it has a lot of amino acids. In aromatic amino acids, we have three: phenylalanine, tyrosine, and tryptophan, okay? Phenylalanine, tyrosine, and tryptophan. So, can you divide these two as phenyl and alanine, okay? Have you heard this before? Alanine, what was the R group in alanine? CH3. Can you hide this structure? Is it a CH3 molecule? Kind of. If you add a hydrogen, it's CH3, right? If you want to add something, you have to remove a hydrogen, right? So this is CH alanine, right? What's the other group you have? It's the benzene. Benzene is also called as phenyl group. Okay, so this is phenylalanine. Means in alanine you have additional phenyl group. Am I clear to that? So we have a phenyl group in alanine, and that's why they are called as phenylalanine. Isn't it interesting how amino acids are evolved in nature? You know, imagine first time they formed alanine, okay, and they want to make a new amino acid, and they added a phenyl group, and they called it phenylalanine. And this is how amino acids are diversified in the course of evolution. Okay. The second one is, remember, you have to is tyrosine. Can you see a very common thing between phenylalanine and tyrosine? Yes. The whole thing is same except this additional OH group or hydroxyl group. Okay. So can we call tyrosine as hydroxylated phenylalanine? Yes or no? Because there is an additional hydroxyl group over there, right? So write down in place of tyrosine, tyrosine is hydroxylated phenylalanine. Okay? So alanine, you know, additional phenyl group, phenylalanine, hydroxylated phenylalanine is tyrosine. Okay? And the other structure you have to remember is tryptophan. Okay? Tryptophan is slightly complex but easy to remember, okay, once you know how to draw the structure of tryptophan. But you have to repeat after me, okay? Okay, let's see what is common between these three. The common between these three is a phenyl group, okay? I think you can easily identify phenyl group over here and here, but there's also hidden phenyl group over here. Can you see these three double bonds? They are in conjugation and they are aromatic. These molecules are called aromatic molecules. They have a property of aromaticity, okay? And the only thing you have to remember is 1, 3, 4, 1, 2, 3, 4. I told you, fourth position is very important to remember the structure of amino acid. And in fourth place you have nitrogen in place of carbon. Okay? So 1, 2, 3, 4, and then a benzene ring. Okay? So you can remember 1, 2, 3, 4, and then a benzene ring. The additional double bond will be opposite to this double bond. Okay? You have to draw maybe a couple of times to remember the structure of amino acid, but it's very simple, right? The best way to remember or draw the structure is draw your carbon chain first, okay, which is basic structure. Hydrogen, hydrogen, COOH, hydrogen, NH2, then a CH2 group. The CH2 group is common in all of them. Can you see that? Because they are all almost the similar form of alanine. Okay. The other thing you have to remember is 1, 2, 3, 4. Okay, and then a benzene ring. draw this benzene ring first if you are confused anyway, and draw a double bond in parallel to this. That's it. This is how you remember tryptophan. This is one of the most complex amino acid you have to remember, but it's very easy. Why? 1, 2, 3, 4. Fourth position is nitrogen, everything else is carbon. And you have a benzene ring, opposite to your benzene ring there is a double bond. That's it. Okay? So alanine is CH3, phenylalanine is CH3 plus a benzene ring or phenyl ring. Okay? Tyrosine is hydroxylated phenylalanine. Okay? And tryptophan is 1, 2, 3, 4. Fourth position is nitrogen, everything else is carbon. And you have a benzene ring, opposite to that you have a double bond. Okay? Too many information to mug up, right? But you have to practice at least 2-3 times to remember structures, right? But you will be fine. I promise you that. Okay? We are done up to here. Any questions? Any doubt up to here? No? Cool. Let's move ahead. Very easy. Let's come back to Apple again. Okay? You have two giants in United States in terms of technology, right? One is Google, other is Apple. Okay? You know the CEO of Apple? Anyone? Steve Jobs is dead. He was an excellent entrepreneur. I love him. Anyone? You should know. You are young, you know. No? Anyone? CEO of Apple? Tim Cook? You heard that name? Tim Cook? I think he's a bald guy. Okay, like me. And Apple is? Anyone? He's an Indian. I'm proud of that. Sundar Pichai. Okay? He's a Apple, Google CEO. Okay, why I'm talking about all that? Because you have two giants, Apple and Google. This is asparagine and this is glutamine. Okay? And you have to learn something and get into Apple or Google, right? And what you have to learn? Technology, coding, computers, right? So I would say you have to learn computer science, which is C and S. Okay? And computer science will lead to Apple and Google. So CS2 A and G. Are you getting my mnemonics or no? It's funny. No sense. Okay? It's a nonsense. But this helps. This is how I used to find tricks out, remember, or there's a system how to click your memory to another thing. Okay, this is called hooking system. Okay, if you already know something, hook another thing to that so that you already remember along with that. Okay, this is a very good practice to remember things. Anyway, that helps to me, and I think it will help you guys too. Okay, I'll say computer science to Apple and Google. Right, C for cysteine, S for serine, T for threonine, A for asparagine, and G for glutamine. Okay, let's start with C. C is cysteine. Okay, it's very interesting. Number 3 is important now. Earlier, number 4 was important. Now, number 3 is important. Why? Let's see. 3 means threonine. 3 means threonine. And you have 1, 2, 3. One is OH, another is carbon, another is CH3. Okay? Before that, let's move to serine. Okay, serine and cysteine has something in common. And again, there's an evolutionary trend you will see. Can you see the R group you have? R group is highlighted or said over, right? R group is CH2OH. Right, CH2OH. CH2OH is hydroxyl group, okay? And cysteine is very similar, CH2SH. So see how smart nature is, okay? He replaced oxygen with sulfur, and now they have a different amino acid as cysteine, okay? So serine and cysteine, they are sisters, right? Sister amino acids. They have oxygen replaced by sulfur, okay? And threonine is 3. You have to remember 1, 2, 3, OH, CH, and CH3. Get that? The other two you have to remember is asparagine and glutamine. I don't know where is competition, but there is one CH2 and there is two CH2. A and G. You can remember, like A is first alphabet and G comes afterward. So you can say this is A for 1 and G for 2. Okay, one CH2, two CH2. And the other thing you have to remember, they have an amide group which is CO and H2. Okay, which is common. Okay, asparagine and glutamine. Why? They have amine. They have amine. That's why they are called as asparagine or glutamine. Last amine. You see that? Asparagine and glutamine means they have an amine group. They have an amide group. Okay. Amide group is common, one CH2 and two CH2. That's it. Okay. Let's start with cysteine. No, start with serine. Serine is CH2 OH. CH2 OH. Cysteine is CH2 SH. Now I'll ask you a very simple question and you know that. You know the answer. You know the answer. What's your name?
Rahul.
Rahul know the answer. What is, can you give me an example of sulfur containing amino acid? Methionine, exactly. I told you, you know the answer, right? So we have another sulfur containing amino acid which is cysteine. Okay, and now we have two sulfur containing amino acid. One is methionine and other is cysteine. Cysteine and methionine are sulfur containing amino acids. That's it. Full stop. Right? Cysteine and methionine are sulfur containing amino acid. Okay. So you remember serine and cysteine now. Threonine is 3. 3 is OH, CH, CH3. Right? Threonine. Now asparagine 1, glutamine 2. 1 what? CH2 and 2 CH2. Amine means they have a CO and H2, which is a amide group. Okay? Can you remember that? You can make small notes in order to remember things. Okay? We are done up to here. Any questions? No. Serine is, let's speak up with me. Serine is CH2 OH. Cysteine is CH2 SH. Threonine is CH OH CH3. Right? And asparagine CH2 CO NH2. CH2 CH2 CO NH2. Glutamine. One more time. Serine is CH2 OH. Okay? Cysteine CH2 SH. Okay? Threonine CHOH CH3. Okay? And then asparagine CH2 CO NH2. Glutamine CH2 CH2 CO NH2. That's it. Okay? Up to here we're clear. One more time and we're done. Hold on. Third one is last. This is last one, and this is positively charged amino acids, right? And we have lysine, arginine, and histidine. Okay, I think this is helpful, right? What do you think? Is it okay to remember like structures of all the amino acids you have? So this is lysine, arginine, and histidine. Okay. In your body, there is a very good system of LHA complex. HLA complex, sorry, HLA complex, which is human leukocyte antigen. Okay, have you heard of tissue grafting, grafting or organ transplantation? You know, people donate their kidney, heart, eyes, or anything else, and then it goes to another body, which is recipient. And I'm a donor if I donate any body organ, and they are capable of functioning the same in other body. Okay, but there are high chances of rejection. You know, doctor says sorry. Body is not accepting the organ, right? And that's a tissue rejection, okay? And they have a complex called HLA histocompatibility complex, okay? That's how they recognize. I recognize this because I have a biology background and do biochemistry a lot, okay? HLA. Okay, but it's very common, HLA system, okay? So let's remember what is common here is 4 carbon, 1 nitrogen, okay? 4C plus 1N. That's common for remembering positively charged amino acids. Okay? 4 carbon, 1 nitrogen. How? Can you see this? 1, 2, 3, 4. 4 carbon, 1 nitrogen. That's it. 4C plus 1N. The other way to write this is 4 carbon and 2 nitrogen. 1, 2, 3, 4. No, this is 2 nitrogen. 1, 2, 3, 4, 1, 2. Okay. This one is 4 carbon, 1 nitrogen, 4 carbon, 2 nitrogen, and 4 carbon, 3 nitrogen. That's it. Okay. But the way to remember this is, first is LHA. Okay, let's remember this as LHA because it will be more useful then. Okay, how you remember that? Write LHA. Okay, and then LHA 4 carbon 1 nitrogen, 4 carbon plus 2 nitrogen, and 4 carbon plus 3 nitrogen. Right? Now, how you can arrange 4 carbon 1 nitrogen is simply put 4 carbons one after the other. 1, 2, 3, 4, 1 nitrogen. Okay? You have to remember this. And I think this is very similar to another amino acid structure. Anyone? Tryptophan. See that? So there. See that? These rings look very familiar, right? But they don't have an aromatic group. Is there an aromatic group? No, that's why they are not categorized into aromatic amino acids, right? They are categorized into positively charged amino acids. So the positive charge, can you see that? This one positive, and this one is also positive. Okay? 4 carbon, 1 nitrogen, this is 4 carbon, 2 nitrogen, histidine. So two most complex structures you have seen is one histidine, other is tryptophan. Right? Tryptophan and histidine are very similar but complex structures. Okay? These are only two complex structures you have to remember. And they look very similar. So don't get confused. You have to practice in order to remember they are different. Histidine is not aromatic. So there is no aromatic ring or no phenyl ring. The other you have is tryptophan, and they have an aromatic ring. Okay? Again, if you have 2 nitrogen, okay, you cannot put 1 over here. You remember the other structures you remembered for tryptophan, it has nitrogen in the 4th position, right? Now you don't have a nitrogen at 4th position, but you have a nitrogen at 3 or 5th. 1, 2, 3, 4, 5. You see that? 3 and 5. If it's a tryptophan, the nitrogen will be over here in the 4th position. If it's not tryptophan, nitrogen, everything is same except these two bonds are parallel to each other. You see that? Slight difference, but after practice you will be able to remember, right? So again, 4 carbon, again you have to remember 4 carbon, 2 nitrogen. If you have 2 nitrogen, you will place it one after the other over here with a bridge of carbon, right? So 1, 2, 3, 4 and 2 nitrogen, okay? 2 double bonds over here. Can you see that? If it's a aromatic amino acids, this double bond will shift over here, right? Here and here, and there is a benzene ring, and that's it, right? So this is how you remember. The last one is arginine, okay? Arginine is simple, but you have to remember there is 3 carbon, okay? Let's 1, 2, 3 carbon, no, 4 carbon, right? 4 carbon and 3 nitrogen, right? So 1, 2, 3, put a carbon in the middle and surround by 3 nitrogen. Okay? And balance the hydrogen accordingly. I'm not talking much about hydrogen, right? Because it's a chance if they are present in acidic or basidic medium. Okay? It changes. But 4 carbon, 1 nitrogen, 4 carbon, 2 nitrogen, and 4 carbon, 3 nitrogen. Is it possible to remember now? Yes, sir. Yes or no? Okay. So 1, 2, 3, 4, 1 nitrogen, 1, 2, 3, 4, 2 nitrogen, 1, 2, 3, 4, 3 nitrogen. Okay? Clear up to here? And there's a double bond. You don't forget. You should not forget the double bond, right? This one is parallel The opposite you have to remember is for tryptophan, it's this parallel. Okay? Horizontal parallel, vertical parallel. Okay? Cool. That's up to here. The last one is very similar. So you already know that. Apple and Google, they come back. Right? Apple is 1CH2 and Google has 2CH2. I have a very concerned request, do not write Apple and Google when you have a exam paper, right? Two negatively charged amino acids, Apple and Google. Sounds weird. Aspartate and glutamate. You can call this as aspartic acid and glutamic acid if you add a hydrogen over here and here. Aspartic acid and glutamic acid. If you don't have a hydrogen, this is an ion, and that's why they are called as aspartate and glutamate. Okay? But if you add a hydrogen, this is aspartic acid and this is glutamic acid. Okay? Clear up to here? Any questions? Anyone? No. Apple has one. Since this is an acid, you expect a carboxyl group, right? Because acid has capacity to lose hydrogen, and your acidic group is carboxyl group. One over here, one over here. That's it. One has 1CH2, and the other is 2CH2. Okay? Let's. How many times? How much time we have left? 20 minutes. Okay. I'll give 5 more minutes to remember, 3 minutes. Okay. To start from the start, from the very first group we have. Okay. And let's see how much you still remember. Right. Let's move off to over here. Okay. So you can close your notebook and focus here, right? The first mnemonics that we have remembered is give me apple, right? Apple and Google, everything surrounds the amino acid, right? So first is glycine, which is simplest. Okay, glycine is H, alanine is CH3. Okay, glycine, alanine, and then we'll talk about leucine. Okay, leucine is 1, 2, 3, 4. Okay, isoleucine is isomer of leucine. Let's bring that over here. CH2, CH3, CH2, CH3. Right. This is isoleucine. Valine is opposite B. CH, CH3, CH3. Right. Give me for methionine, and methionine is sulfur containing amino acid. Right. And CH2, CH2. Sorry, it's CH2, CH2, S and CH3. Okay. So third position is sulfur, and everything is carbon, right? And then A for alanine, I think we already covered that. P for proline, proline is circular, 1, 2, 3, 4, 4 ring structure. And then L for leucine, I think we already discussed that, right? Clear. H for lysine, CH3 for alanine. Leucine is CH2, CH2, CH3, CH3. Isoleucine is CH2, CH3, CH2, CH3, right? And then valine is opposite B, CH2, CH3, CH3, and then methionine is CH2, CH2, S and CH3, right? And then proline is ring structure, 1, 2, 3, 4 ring structure, and then I think we already discussed about leucine, right? So let's move ahead and see aromatic amino acid. Anyone remember phenylalanine? It's an alanine with a phenyl group, and then hydroxylated phenylalanine is tyrosine, and again tryptophan is another amino acid with tryptophan. You remember that? Tryptophan is 1, 2, 3, 4. Fourth is, come on, fourth is 1, 2, 3, 4. Fourth is, everything else is carbon. Okay, there's a benzene ring, and opposite to benzene ring is a double bond. That's it. Okay, move ahead with the next. First is serine. Serine is CH2OH. Okay, and cysteine is CH2SH. Threonine is 1, 2, 3 CHOH CH3. Asparagine, apple has 1 CH2, and glutamine has 2 CH2. And apart from that, they have an amide group CONH2, CONH2. Same? Clear up to here? Any questions?
What did you call that? An harmonic group?
This one, asparagine.
The C double bond and then the amine attached to it?
Amide.
Amine.
Okay. And then finally LHA, which is lysine, histidine and arginine. 4 carbon, 1 nitrogen. 4 carbon, 2 nitrogen. 4 carbon, okay. And the similar structure is 4 carbon, 1 nitrogen. 1, 2, 3, 4 carbon, 2 nitrogen. 1, 2, 3, 4 carbon, 3 nitrogen. Okay, that's it. And then this is the brother of asparagine and glutamine. But this is aspartic acid and glutamic acid, which is aspartate and glutamate similarly, right? And they have 1 CH2, 2 CH2. That's it. Simple and easy. Simplest thing to remember. But you have to practice maybe 3 times, and I guarantee you can teach anyone how to remember the names of 21 amino acids you have. Okay? Clear? Any questions up to here?